This is Huang from Mount Sinai, and today I'd like to talk to you about my previous work, a common haplotype lowers PU.1 expression in myeloid cells in the late onset of Alzheimer's disease. This was work published 2017 in Nature Neuroscience, and I conducted multiple work in Dr. Alison Gold's lab before she moved to Mount Sinai, and after she moved to Mount Sinai, we also added on to the work. And this is a highly collaborative work, as you can see, involving co-first author Dado, as well as multiple members of Allison's lab and multiple members of the International IGAP Consortium and experts at WashU and Mount Sinai. And let me first provide a little context for this work. By the time we started this project, IGAP has published the biggest GWAS in Alzheimer's disease, reporting around 20 genetic loci associated with Alzheimer's disease using GWAS of around 70,000 people. And obviously, one clear challenge there was that there was not a lot of things known about every gene and each of the loci. And perhaps some of the loci, we don't know which genes are responsible for disease. Another challenge is that then we don't have good ways to really fine map a lot of those loci to find out the responsible genes or responsible variants. And so we started the project by thinking that perhaps we can use more information in the GWAS study. Conventional GWAS study use case control, and that means that in Alzheimer's, you would treat someone who's 50 as a control, but also someone who's 100 as a control. And we feel like there are some age information we could use there because someone who hasn't got Alzheimer's at 50 can still get Alzheimer's, especially getting older. There's a high chance, given the high prevalence of the disease in older individuals. Whereas someone who hasn't got Alzheimer's at 100, that tells us more information. And so we like to introduce this survival GWAS concept into the study. We've also used other type of information, especially CSF biomarker association, and as well as cell type specific EQTL information, which I will go a lot more in depth into. And then beyond that, we then seek epigenomic evidence and in vitro validation evidence for the loci we identify. So let's jump into it. So the paper started out being a more typical GWAS study where we use age of onset in the survival GWAS using the Cox proportionate hazards model, essentially using the IGAP cohort with the age information and doing a meta-analysis of the cohorts. And obviously, we found a lot of previously reported loci. We did not find any new loci that really passed the stringent genome-wide significance criteria, but we reported the ones that have suggestive significance. And for the rest of the paper, we really focus on the one that confirmed with the previous ones. And here you can see almost all the ones were previously reported, but here you can see that the previous reported self-1 locus, our identified loci is closer to the gene SPI1 instead. And we further validated these loci by showing that many of them are associated with CSF biomarker, including tau proteins, p-tau, phosphorylated tau, as well as A-beta. The one that are passing the stringent Bonferroni corrections threshold are then shown in bold. As you can see, that is SPI1 is with A-beta 42, as well as ApoE with p-tau and A-beta 42. And after that, what we did was we looked into where any of these loci showed evidence of EQTL in the brain region, and we did not find much there. The result is reported. And so at that time, the field has this intriguing evidence that many of the new Alzheimer's genes, such as ThyroBP, which is identified in Dr. Bingsong's paper in Cell, as well as TREM2, which is identified in sequencing study, are all more likely microglia expressed in the associated genes. And so we might, instead of finding Alzheimer's genes that may be playing their role in neurons or astrocytes, we might think about looking more into the immune signal. And so indeed, when we did LD score regression, that is what we found, where the the genome-wide association study significant signal does overlap with immune cell EQTL landscape better than it would have than, say, schizophrenia GWAS data. And that really intrigued us to look into whether any of these loci are correlated with EQTL in immune cells such as monocytes and macrophages closer to microglial lineage. Obviously, microglial EQTL data is really hard to come by. So when we do that, we found some very intriguing strong signals. So as you can see, some of the strongest signals are really in besides from cell 1 in chromosome 1, PIGG in chromosome 4, a lot of strong ones are in chromosome 11, including SBI1, which has the strongest p-value, very significant in both monocyte and macrophage. We also found very strong signal for MS4A4A, MS4A6A, the identified genetic risk loci, or here the genetic risk loci associated with survival really has stronger association with these genes in the immune cells. And so I think this provides really strong evidence, especially for the SBI1 loci, that these genes potentially are more likely to be the real signal than, say, previously reported SELF1 that show association in the previous GWAS, or closest to the association in the previous GWAS. And here, just diving into it a little more, you'll see that the survival association GWAS landscape looking like this, which is quite different from the case control IGAP study where everything is much more flat here, is really close to what the monocyte SPI1 EQTL or macrophage SPI1 EQTL looks like. And the same thing we observe for the monocyte EQTL for MS4A4A at the other loci. And so obviously, we did statistical tests on this. Co-localization of these signals at these loci are very, very good. And that gives us further confidence that potentially the contribution of these loci to Alzheimer's, to survival without getting Alzheimer's disease, is really driven by transcriptional regulation of these genes. And so to further support, especially the evidence at the SBI1 loci, here is just what the different genotype of the SNP were identified to be most strongly associated with AD survival to SBI1 expression. And you can see that when you have two alleles, you have much lower SBI1. And here we confirm that SBI1 is indeed a gene expressed in microglia instead of other neuronal cell type. And so these really, again, reaffirm our hypothesis that this SNP is really driving differential expression in SBI1, which is expressed in microglia. We then look into how SBI1 may assert its effect functionally. So SBI1 includes codes for PU.1, which is a well-known myeloid lineage transcription factor. And so we kind of hypothesized that it will bind to various genes. And indeed, we see enrichment of PU1 binding in a series of immune genes. And here we're showing some examples. So if I zoom in a little bit, here, for example, we're looking at the genetic loci at CD33, which is an Alzheimer risk gene. And here are the tracks for PU1. You see that PU1 clearly binds to the promoter area of CD33. And you see that as well for MS4A6A. Here, you see the PU1 signal here, and you see that for MS4A4A. You even see that very intriguingly for potentially TREM-L2. And maybe TREM2, but it's not really in the promoter, it's in the 3' UTR region. And so This gives us really convincing evidence that PU1 can be this master regulator that modulates the expression level of all these other microglial genes that all are known to play a role in Alzheimer's disease. And we provide in text more information supporting this hypothesis. Given the strong signal we observe for PU1, we further carry out functional validation. And this work are really an effort by Dado and Anna in Allison's lab, where first we introduce overexpression of PU1 as well as gene expression knockdown of PU1. And you see that it clearly affects the microglial cells, mice microglial BV2 cells, phagocytotic activity, where the overexpressed cells have stronger phagocytotic index than the knockdown one in a very significant manner. And here, we also again do the SPI1 or PU1 overexpression and knockdown and see that if it does affect the other Alzheimer gene as we have identified them in the promoter binding sites. And so, for example, we see a significant difference of the MS4A4A and S4A60, the mice monologue of those genes affected by SPI1. Intriguingly, we also see other secreted molecules as well as lipoproteins, the Alzheimer's risk genes ApoE and CLU, being differentially expressed after SPI1 or PU1 overexpression or knockdown. And we see that again for more genes on knockdown only, including CD33, thyroid BP. And so this really gives us a stronger causal relationship of PU1 expression level modulating these key Alzheimer's genes, and that may in turn affect Alzheimer risk. And so overall, we believe our study gives a really convincing evidence how PU1 may be a master regulator microglial gene that affect Alzheimer's. And this really, again, affirmed the field's thought at that time, especially currently, of microglial's involvement in Alzheimer's disease. With that, I'd like to thank you